The molecular biology session, sponsored by Novartis, is about to begin. Our speakers for this session are Professor Kiwon Ban and Dr. Sai Chavala. The shared person is Professor Perpetua Pinto Duol, President of the Portuguese Society for Stem Cells and Cell Therapy, as well as Senior Investigator at I3S and INEB, leading the research group focused on stem cells and regenerative biology.
Good afternoon. Welcome back from lunch. I hope you have restored your energy for this afternoon session. It is my great pleasure to introduce the first speaker of this session, Professor Kiwon Ban. Professor Kiwon Ban, main topic research is in cardiac repair. He is presently at University of Hong Kong, where his group is focusing in different approaches toward cardiac repair and one day regeneration. This is eventually for later discussion in the session. Professor Kiwon Ban did his bachelor and master degree at the University of Dongkung in Seoul, Korea, and he moved then to Canada, where he did his PhD in Toronto at the University of Toronto and at the Department of Physiology. And soon after that, he moved for his postdoctoral studies to the United States, to the Department of Medicine at the Emory University. Very prestigious place as well in regenerative medicine. Following what Professor Kiwon Ban went to Hong Kong, where he is presently, and from where he is talking to us into the how are we going to repair cardiac disease and injury in future. Thank you so much for being at this meeting with us today.
Hello, everyone. My name is Kiwon Ban at the City University of Hong Kong. So it is really unfortunate that I cannot attend the conference and give my presentation and discuss my study in person. However, I try my best to introduce this one of my recently published study and deliver my message as clear as possible through this pre-recorded movie. So as you know, heart failure is one of the leading cause of death worldwide. And especially this late stage of heart failure, the patients, so they don't have such therapeutic available option except this heart transplantation and artificial heart device. So, however, these two options, heart transplantation, have lack of donors. And also, this artificial heart device have several side effects and disadvantages, so that these are not really realistic and promising option for this late stage of heart failure patients. So in this regard, this in last 20 years, stem cell-based cardiac regenerative therapy has received lots of attention as a promising and alternative option for this late stage of heart failure patients. So our unique views for true cardiac regenerative therapy would be because this heart failure or myocardial infarction excessively damaged both myocardium and then cardiac vessel, which are two major components in the heart. So therapeutic strategy for treating this failing heart should concurrently target both myocardium and then vasculature to achieve comprehensive cardiac repair. So to regenerate cardiac muscle, we employed cardiomyocyte derived from the human pluripotent stem cell, which including both human embryonic stem cells and human-induced pluripotent stem cells. Because these cardiomyocytes derived from the human PSC have a largely similarity with the human primary cardiomyocyte in terms of structure, and then cardiac gene expression, and cardiac specific protein expression, and then electrophysiological characteristics. And more importantly, these cardiomyocytes show spontaneous contraction over time. And then when they implant into the heart, they set up the junction together with host myocardium through the expression of gap junction protein connection 43. And then they show the synchronous contraction with host myocardium, and then ultimately improve cardiac function. So these are cardiomyocytes derived from the human PSC on day 14, and they show the continuous spontaneous contraction over time. They also form the aggregate, the cardiac organoid. Another name is cardiac body, and consistently show spontaneous contraction over time. And these are the magnified image also show the continuous contraction. In addition, to regenerate cardiac vessel, among those many cell candidates of stem cell, we have chosen the human mesenchymal stem cells from the bone marrow. It is because those human mesenchymal stem cells, although they didn't show significant improvement in cardiac function through these multiple human clinical trial, they consistently show significant effect on angiogenesis, suggesting that this human mesenchymal stem cell have an effect on vascular regeneration. So our strategy would be we inject through the human PSC-derived cardiomyocyte directly into the heart, intramyocardial injection. And then we made human mesenchymal stem cell patch, which encapsulating human bone marrow mesenchymal stem cell, and produced the patch through the 3D bioprinting with pig heart-derived extracellular matrix to allow them consistently secrete several beneficial paracrine factors from this human mesenchymal cell patch. So our hypothesis would be human mesenchymal cell patch can increase vascular regeneration through the prolonged secretion of beneficial paracrine factor, which including angiogenic cytokine. And then those intramyocardial injected human, the PSC cardiomyocyte ultimately enhance cardiac function. So as a first trial, we generate a human mesenchymal stem cell patch through the decellularized heart tissue. So we simply purchased the pig's heart from the conventional slaughterhouse, and then we applied decellularization, and then lyophilization and digestion to produce the HD ECM bioink for conventional usage, storage, and then application. And then we eventually produced the human mesenchymal stem cell patch through the 3D bioprinting technique and encapsulating human mesenchymal stem cells. So these are a finer shape of human mesenchymal stem cell patch. So to confirm this human mesenchymal stem cell patch is biologically active and functional, we produced the HMSC patch and then cultured them in vitro condition for several days. And we harvested the supernatant from the culture media and then analyzed through the BASEP enzyme kit, the ELISA kit. And finally, we confirmed that this batch, the human mesenchymal patch, secrete significant amount of the VEGF, which is the, one of the major components in angiogenic cytokine. And this show that on day 3, they have increased expression of VEGF on in vitro condition, suggesting that this human mesenchymal patch is biologically active and functional. And ultimately, we implanted this human mesenchymal stem cell patch in the rat, which induced the myocardial infarction through the ligation of the LAD, which is one of the coronary arteries, and implanted this patch outside of the heart through the epicardium. And then we expected this patch can secrete the beneficial cytokine over time. As a result, this, the heart received the patch post-MI the first week, and we harvest them and then analyzed through the QRT-PCR analysis and found that this patch really improved several genes associated with the angiogenesis, such as VEGF, IGF-1, and FGF2, ANG1, and HNG2. So all these genes are significantly upregulated through the patch generation in this heart received the patch compared to control group. So suggesting that this patch really improve the angiogenic genes in MI-induced heart. And ultimately, we collected the heart received the patch of human mesenchymal stem cell and then measured the number of capillaries to evaluate the effect of the vascular regeneration and show that this patch in the attached heart have a significantly higher number of capillaries in compared to control MI heart, suggesting that this human mesenchymal stem cell patch really improved the vascular regeneration in the MI-induced heart. And also, this dual approach with the cardiac myocyte and the mesenchymal cell patch really improved the cardiac function through the evaluation of the heart function of the regular echocardiography checkup. So, as this bar graph show that this dual approach, CM plus PA, in two categories, the ejection fraction and then fractional shortening, show that this dual approach treated heart have a higher heart function in compared to cardiac myocyte alone injection show as a red bar, and then control MI heart show as a gray bar, suggesting that this dual approach substantially improved cardiac function in MI-induced heart. So one of the interesting findings from our dual approach is that this dual approach dramatically increased the retention of intramyocardially injected the cardiac myocyte. So as you can see in these two images in the cardiac myocyte alone injection and then dual approach. So those pre-stained cardiac myocyte with the red dye show that number of cardiac myocyte remained engrafted, and number is significantly higher. This cardiac myocyte alone injection group only minimal, a very small number of cardiac myocyte remained over the myocardium. However, this dual approach, the red dye stained cardiac myocyte is a very equally distributed in the all over the myocardium, and the number is significantly higher. As you know, this one of the critical barrier for stem cell based therapy is low retention and then engraftment. So previous study showed that almost 50% of injected cells are lost immediately, and also after 1 week the treatment, only 10% of the injected cells remained and survived, suggesting that this poor survival of injected cells into the ischemic heart is a very critical limitation to overcome for cell-based cardiac regeneration therapy. So our dual approach, as you show in this two image, and then those quantification over these three sections in the heart in different group, show that this dual approach, CM plus PA, significantly improved the number of the cells remain in the heart tissue compared to CM-only group in the red bar. So this image, we can easily distinguish this cardiac myocyte in the dual group and then CM-only group have a huge difference in terms of remained the cardiac myocyte, which show the GFP. So another interesting finding from our study is that we dual approach really promoted the maturation of injected cardiac myocyte. So as you see, clearly this cardiac myocyte only injection group and the versus this dual approach, the cardiac myocyte with the patch, show that this only group have a very the small in size and then very irregular shape. And importantly, it's very small, right? First of all, and the size is much larger in this dual approach group, and they contain very organized the sarcometic structure in this injected cardiac myocyte, suggesting that this dual approach promoted maturation of injected cardiac myocyte. So in our field, it has been very well known that those cardiac myocyte differentiated from the human PSG cells have a very immature phenotype. So as you show in this figure, this PSG and early HPSGM and then late PHGM, in terms of comparison with the organic structure and then shape and then the size is very different. So this adult human mature cardiac myocyte very well organized sarcometic structure. However, although this late stage, which we considered the pretty much matured over the time or through the many application to promote maturation, still this late the HPSCM and then other human CM has a very clearly different in parameter for distinguishing maturation of cardiac myocyte. So our study showed that this cardiac myocyte, first of all, the human cardiac myocyte injected, to confirm the human protein expression, human myosin heavy chain, and human mitochondria confirmed that this GFP signaled human cardiac myocyte is really human. That their identity is human. And then this cardiac myocyte is the GFP displayed the cardiac myocyte is really compared to cardiac myocyte only injection, very different with the size. And then if you can see this clearly, they have a very well organized sarcometic structure, suggesting that this human the dual approach really improved the maturity or maturation of cardiac myocyte. So actually, this is an image I took when I was postdoc, 2014, published in ACS Nano. So even at the time, I really considered that this cell therapy with PA-RDDS biomaterial really improved the maturation. However, compared to this image I captured by myself in 2014, and then the image I captured in 2019, really huge difference. So this is evidence that this our dual approach really promoted maturation of the cardiac myocyte. So and also one of the evidence of the mature cardiac myocyte is a junction with the host myocardium, as I introduced earlier. So GJA1, another name is connection 43. So in this cardiac myocyte alone injection group, they don't have such expression of the GJA1 with the host myocardium and then cardiac myocyte GFP cardiac myocyte itself. However, this cardiac myocyte injected through the patch, like dual approach, really improved the expression of the GJA1, which showed a gray color. And then they have a conjunction with the host myocardium in this, the overact image and then the lead image of host myocardium. And also this here, it's very abundant expression of GJA1 and have a junction with host myocardium. Also, additionally suggests that this dual approach really promoted the maturation with the expression of junctional protein and set up the junction with host myocardium, and then eventually improve the cardiac function through this mechanism. So as an underlying mechanism, we found that this human mesenchymal stem cell are very survived over the time, so until 8 weeks. So those red dye eye image, which pre-stained with the human mesenchymal stem cell before implantation, suggesting that these human mesenchymal stem cell are very happy and survived well, living well within the patch and after implantation into the MI heart until 8 weeks. And then they secrete a significant component of several cytokines related to angiogenic gene, about 30.1%, and then immune regulation and cell viability, and then ECM demodulating, which really involved in survival of injected cardiac myocyte and cardiac remodeling survival, right? And also, we also found that several, the significant expression of DKK1 and DKK3, which are Wnt inhibitor that involved in maturation of cardiac myocyte. So these, those protein or cytokine persistently secreted from the human mesenchymal patch really improve the retention, survival, engraftment, and then maturation of cardiac myocyte injected directly to the heart, and then those patch really contribute their survival and maturation. So as a summary, we found that it's our dual approach through the intramyocardial injection of a human PSGM and then patch of a human mesenchymal stem cell really improved the cardiac function and reduced the cardiac fibrosis and improved the vascular regeneration and retention and engraftment of cardiac myocyte. And finally, those survived and remain in the heart tissue really improved maturity of the cardiac myocyte after transplantation. So thank you very much for listening to my talk. And unfortunately, I cannot have a Q&A, but if I have a chance later, we can discuss in detail later. Thank you very much.
Thank you so much. Thank you. Thank you very much, Dr. Kiwon Ban. It was a very clear and very exciting lecture. I believe we all appreciate it very much. So we will now go straight away without further interruption to our next speaker, Professor Sai Chavala from University of, from Duke, sorry, I was actually in his beginnings a long ago. From the Texas Christian University, University of North Texas Health Science Center, School of Medicine in Texas. Dr. Sai has done his medical degree in the beginning of this century. He's young, though. And he did his residency in ophthalmology 15 years ago, from where he proceeded to a postdoctoral research program mentored by Professor Shaheen Rafi, a reference person in the stem cell field and in the angiogenesis field, I would say. And from where he moved from surgery specialization in Duke University to his present position in Texas. He actually accumulates the duties of a chief medical officer with that of being a professor at Department of Surgery and more recently an executive director for the Center for Retina Innovation, a nonprofit research and development unit in retinal disease. We are very eager to listen to what Professor Sai Chavala is going to tell us. Very welcome to this session. Thank you for accepting the invitation. The stage is yours.
Thank you very, thank you very much for the very kind invitation. I'm very, very excited to be here. As I understand, the meeting was student founded and student powered, which makes it even more exciting. My presentation really is about a journey. And as you very kindly described, I'm a physician. And so the journey for me was really to start out by caring for patients that had blindness or disease that led to vision loss. And that was always my my aspiration, was to directly care for patients. And during my residency, I had the great fortune of having research labs very close to the clinics. And I started to dabble in research. It was really more of an experiment of sorts. And I realized very quickly that I could impact millions of people by doing research that could potentially be translated to clinic. And so that has always been my singular focus, is to use basic science research to try to translate the newest therapies for those that don't have any therapies right now. And so I'm very, very excited to speak to you about my presentation. We were fortunate to have a recent publication in the journal Nature that highlights our work. And without any further ado, I'll start to present. My financial disclosures are that I am the executive director for the Center for Retina Innovation, and I also have an equity interest in Circ Therapeutics. This is the technology that you're going to see, and it was licensed from the University of North Texas that has intellectual property regarding the technology. So for those of you who are not familiar, the eye is an extremely small organ, but it's extraordinarily valuable and exciting for scientific research because it is easily accessible. And the cornea, which is the clear part of the eye, is optically transparent to allow light rays into the eye. And those light rays focus on the back of the eye called the retina. And the retina is the outpouching of the central nervous system that allows us to convert light into an electrical stimulus, which the brain then perceives as vision. What is very important is the macula. The macula is the central part of the retina. This is the part that is responsible for 20/20 vision, central vision. And the peripheral retina is responsible for our peripheral vision. So this is a retinal image. This can be obtained in our clinics within seconds. It's very, very simple. And we can get a tremendous amount of information in a real live human being about not only the eye, but also about systemic health. So this is the optic nerve. So this is the nerve that takes all of the information that is processed in the retina back to the occipital lobe of the brain and allows us to perceive the world. These are the retinal arteries. And so we can get not only information and data about the retinal vasculature, but also how the systemic vasculature is doing. And some of you may be aware that Google and some others have used artificial intelligence to look at the caliber of the retinal vessels to make very exciting extrapolations about high blood pressure and diabetes from this data. And this is the macula. This is the area for central vision 20/20. So again, this is a very normal, healthy retina. But the problem is that there are patients that are born with a genetic mutation that leads to retinal disease later in life. So these are individuals who can have autosomal dominant, recessive, X-linked genetic mutations in the photoreceptors, which is the important neuron in the retina that takes the light cells, the light from the outside world, and converts it to an electrical stimulus. And over time, these patients that have these genetic defects will develop these bone spicules, and you can see this black pigment within the retina. This is not normal, and this is actually a fibrosis response from the underlying retina, the retinal pigment epithelium, that is showing us that the photoreceptors are dying. And so they undergo apoptosis, usually in the second, third, or fourth decade of life. And this can progress to complete blindness. Now, unfortunately, the issue is that there are more than 60 genetic mutations that cause retinitis pigmentosa, and there are many, many more that cause inherited or genetic causes of retinal degeneration. These start out by patients first noticing tunnel vision. They can actually notice night blindness, where at night they have difficulty navigating, and then it can affect 1 in 3,000 people. Now, the issue is, and again, what inspired me to go into basic science was that these are patients that have no FDA-approved therapy. So, it was very frustrating as a physician to see these patients and then tell them, I have nothing for you. I have nothing that I can offer you. And that's really what inspired me to go into this line of research over more than a decade. So what's really happening? Well, the genetic mutation in the photoreceptor causes photoreceptor cell death. And so over time, as these cells function, they undergo stress, and they eventually undergo apoptosis. And so what is very, very important and interesting is that the neural circuits that are distal to the photoreceptor actually remain intact, meaning that the bipolar cells, the ganglion cells, and the pathways to the occipital above the brain stay intact, but it's just the photoreceptor cells that die. So it's very, very exciting to think about a notion where stem cells could be coaxed into retinal cells, these photoreceptor cells that are here, and that these photoreceptor cells could then function to restore vision. And this paradigm is really what 99% of stem cell biologists around the world are using, is the fact that they can use embryonic stem cells or iPSCs and differentiate those cells into photoreceptors or any type of cell that they desire. So once you get this replacement cell therapy, the question would be, well, then how would you actually restore vision? Well, what's nice is where everything kind of ties together is with my surgical training, we actually have some of these basic principles in place. So we can do what's called a vitrectomy. The vitreous is the jelly in the middle of the eye, and we can actually make ports, small little incisions the size of a very fine pencil. And we can actually remove the vitreous gel and then actually stick needles into the retina and potentially inject cells underneath the retina, and then hope that they actually make meaningful connections to the bipolar cells and restore vision. And so this is kind of a schematic of how that could be done. The idea is that you stick a needle underneath the retina, you would inject your cells, and hopefully those cells would replace the lost cells of the retina related to either genetic causes or acquired dysfunction. And hopefully over time, those would grow and then make synaptic connections with the bipolar cells and restore vision. So about 15 years ago, we asked a very simple question. Are stem cells really needed, and are they the best for retinal pigment epithelial transplantation? And the reason why we asked that question is because embryonic stem cells are fraught with political and ethical considerations, and the fact that they are not specific to the individual warrants the concern for immune rejection. And these subjects may require lifelong immunosuppression with cyclosporin or steroids to suppress the immune system, leaving them susceptible to infections, which is not really a good solution in the long run. So in 2004, as you probably know, Dr. Shinya Yamanaka, who won the Nobel Prize, came up with a system where you could actually take skin fibroblasts, and these could be taken from an individual very simply. It's about a 10-minute surgery where you take a small biopsy of skin tissue, of excess skin. You can grow the skin fibroblasts in vitro. And what Dr. Yamanaka found was that you could take 4 transcription factors that are highly expressed during embryogenesis and topically express them in the skin fibroblasts. and turn them into embryonic stem cell-like cells, which we call iPS. And then you can take those cells, just like you would embryonic stem cells, and coax them into whatever cell type you like. And in our case, these are retinal cells. And that is a really, really exciting notion. But, and this has progressed over the past decade into a therapy that could potentially be used for patients with retinal disease. But the issue is, and you can see this New England Journal of Medicine paper, is that this could take almost 1 year. So for my patients that are blind, the concern would be that they don't want to wait 1 year to have a therapy that may or may not work for them. And so in addition to time, it's extraordinarily expensive to do a manufacturing process with pluripotent stem cells. And also, it is very labor intensive because there's concern that you can develop mutations in the pluripotent stem cells that could lead to tumorigenesis. And so we asked if there was a better way. This came about inspiration from others. So there was a group at Stanford who asked a very simple question: if stem cells were needed at all. And they took the same skin fibroblasts and they used another set of virally encoded transcription factors, and they overexpressed a group of transcription factors and fibroblasts to generate neurons while skipping the pluripotent stem cell stage. And this was published in Nature almost a decade ago by Dr. Wernig. And this was really, really transformational because it would allow us to have a personalized cell therapy approach that avoided stem cells altogether. And we did work related to that, and we used a group of doxycycline-inducible viral transcription factors that we could overexpress in the skin fibroblasts and develop a retinal cell. And so whenever we had LAG-Z positive cells with a RPE or retinal reporter, we knew that we were on the right track. But this also required us using 13 virally encoded transcription factors. So that brought us to reprogramming 3.0. And the question here was, do we really need viral transcription factors at all? Is there a way to pharmacologically convert these somatic fibroblasts into neural cells? And so one of the first papers came out of Science, and this was a small molecule approach to generate pluripotent stem cells. As far as I'm aware, this was only limited to mouse cells. I'm not sure if anyone has replicated this in human. Our group, as well as others, have taken one viral transcription factor and added small molecules and generated pluripotent stem cells. But again, our real purpose was to skip stem cells. And so we wanted to know if there was a chemical approach that could use a transdifferentiation approach to go from skin fibroblasts directly into what we call chemically induced CI neuronal cells. And we were inspired by a group that published 2 papers, one in Cell Stem Cell and one in Cell, showing that this is possible. But this was only limited to generalized neuronal cells. So we use that as sort of the basis of our approach. And we spent years trying to develop a paradigm where we could actually generate retinal cells, so specialized neurons that could actually take this light signal from the outside world and respond to light as a neuron, which we call a photoreceptor. So our approach was simple. We took mouse embryonic fibroblasts from an NRL. So NRL is a very specific gene that is important for rod photoreceptors. And this was tagged to a GFP reporter. And so we took meth from those fibroblasts, and we started looking at different chemical combinations to see if we could generate photoreceptors. And we knew we were on the right track if we had some GFP expression. So our first option was to take the published chemical protocols, and we tried those. And unfortunately, we did not find any GFP cells. We thought, maybe these were groups that were looking at just generalized neurons and weren't aware of the need for photoreceptors. And we thought maybe they had already done it. But unfortunately, those combinations did not yield any types of photoreceptors. So we tried a myriad of small molecule combinations. And ultimately, we generated a 5 small molecule combination that is VCRFI that allowed us to generate these NRL-GFP photoreceptor-like cells. And then we interrogated the need for each small molecule alone. And it turns out that if you subtract out V, C, R, F, or IWR1 or I, that the yield for these NRL GFP cells are minimal. So it requires all of these small molecules in combination to generate a higher yield of these NRL cells. So we did a molecular characterization. So we had these NRL GFP cells that were generated in 11 days. And let me remind you that it took almost 1 year to use a pluripotent approach to generate photoreceptor-like cells. And now we're able to do this in less than 2 weeks. They express the cardinal markers for photoreceptors, CRX. And importantly, we were able to do this in human cells as well. And they expressed recoverin and rhodopsin, which are integral to photoreceptors. And what was really exciting was when we did a non-biased RNA-seq approach. We looked at mouse embryonic fibroblast gene signatures. We looked at rod photoreceptors from mice at P2 and also P5, P3, and adult. And you can see here in our gene signature profile that they look more like a rods that are at the P3 to P5 stage. And why this is important is that there are others who have published a series of high-profile papers in Nature that have shown that the ideal stage for rod photoreceptor transplantation is around P7 to P10. If they are embryonic in nature, the rod photoreceptors don't integrate into the host retina as well. And if they are adult photoreceptors, they do not integrate as well. So they require a certain level of plasticity. And so we were very, very excited that our chemically induced photoreceptor cells, the CIPCs, were around this ontogenetic stage. And that gave us the opportunity to see not only if they resembled molecularly the photoreceptors, but if they could actually function. So we were excited to take 80,000 chemically induced photoreceptor cells, and we actually did an approach that you saw that I described that we could do in humans. We took what are called RD1 or photoreceptor 6 genetic mutant mice. These are mice that go blind by 21 days of age. We have different methods to test their lack of vision. And we injected these CIPCs after 3 weeks, after they were completely blind, and wanted to see if we could restore vision. So one of the most objective tests is the pupil test. And this is a test where you can shine light into the pupil. And if the retina is actually able to have that afferent limb where it can actually take light and convert it into an electrical impulse that then is sensed by the midbrain, it will actually constrict the pupil to limit the amount of light coming into the retina and protect the retina from phototoxicity. So this is taken from our Nature paper, and you can see RD1 is the mutant mice, and these are these 3 columns, and wild type is the far right column. So when we inject saline or PBS into the RD1 mice and shine light, we dark adapt these mice and then shine light, you can see there's no constriction of the pupil. And this is true when we do no injection as well. There's no constriction of the pupil. However, when we inject 80,000 CIPCs, we do get a really nice constriction of the pupil, showing objective proof that the CIPCs are actually sensing the light and making a meaningful objective response in pupil constriction. And we can see this very similarly to the wild type mice here. So this was really, really exciting. And what is also very important is that we do not see a response at 2 weeks, suggesting that we found mice that were truly completely blind at baseline. and at 2 weeks. These were not mice that somehow had less retinal degeneration for whatever purpose. But at 3 weeks and at 4 weeks, when we performed longitudinal studies on the same exact mice, we saw pupil improvement. And this response actually lasted out till 3 months or 12 weeks. So more importantly, the question is behavioral studies. Now that we had objective evidence that these mice could actually sense light, the question is, would their behavior change? Is the brain actually sensing improved vision, and would it change their behavior? So we did a very simple study where we shined light at 50 lux, because rods are responsible for low light or dim vision. And we created a 2-chamber system where the mice could be either in the dark chamber or the light chamber. And it turns out that if mice can see and they're normal, they actually get very scared and they're fearful of the light. They're averse to the light, and then they spend more time in the dark chamber. And you can see that when we objectify the amount of time in the chambers, you can see that wild type mice spend more time in the dark zone. Out of 300 seconds, they spend more time in the dark zone. And then RD1 mice that were injected with our 80,000 cells have a behavior that is very similar to wild type. And then mice that were saline injected, that are RD1 deficient, spend less time in the dark zone. And the reason for that is that they cannot see, they don't perceive the light. So they're not fearful of that, and they can spend time in the light or dark zone, because for them, the light zone is really no light. So for them, both chambers are dark, and they can equally spend time, and they're not fearful. And this is true with no injection as well. One of the questions we had, and one of the hallmarks of chemical reprogramming, and the reason why it's so difficult, is that the question is, once you remove this ectopic stimulus or this exogenous chemical response, do the cells continue to express NRL or photoreceptor genes? And do they survive? And do they integrate? So at 3 months, which is a relatively long time point, and again, a very severely degenerated retina of these RD1 mice, we show that they still express GFP, which was our NRL reporter, and they express this in the right layer, in the outer nuclear layer of the retina, and they actually make synaptic connections with the bipolar cells, which was important. Now, these cells are few. As our previous speaker mentioned, one of the obstacles in cell transplantation, stem cell or non-stem cell, is cell survival. And so we also would note that our cell survival was lower than we hoped. But we did have a meaningful response suggesting that even a minimal amount of cell integration can have a tremendous amount of impact on behavior and vision improvement. And this again is a high-powered image showing that we did have synaptic connections, and which is PKC of the bipolar cells. I know that we are short on time, but please feel free to reference our Nature paper, where we go into great details about the molecular mechanism. I mean, one of the big questions is, how do these 5 small molecules infer a retinal lineage or a retinal pathway? And we go into great detail about how that occurs. But some of the take-home messages are that we induce mitochondrial ROS, and then the NF-kappa B goes to the nucleus and opens up ASCL1, which is a generalized, powerful proneural transcription factor. And ASCL1 turns on CRX and OTX2. So is this clinically relevant? Well, if you look in our paper, one of the major issues is that we only generated 0.9% of NRL GFP cells in the mouse. And less than 1% is not ideal, but it is consistent with other chemical reprogramming methods. And we note in one of our supplemental tables that in human, when we migrated from mouse to human, we were able to show that this is applicable to many different healthy human donors at a wide variety of ages. And we were also able to reprogram some patients with retinitis pigmentosa. So we were able to reprogram their skin fibroblasts. This is exciting for an autologous approach because one could potentially genetically correct the retinitis pigmentosa genetic mutation in the fibroblast stage with gene editing tools, and then chemically reprogram them into photoreceptors and have competent photoreceptors that are specific to that individual. So you could have a precision medicine approach. And then when we modify the protocol, we were able to get this up to 24.9%. So what we have found is that each cell source requires protocol optimization, but the efficiency can be improved. And what's also nice for a clinical therapy for the retina is that, remember, I had mentioned that the macula is what we care about the most. And it turns out that you only need 1 million cells or 1 million photoreceptors to really restore vision. So this is very achievable because fibroblasts are abundant and they grow readily. And so it's very easy to achieve 1 million cells, even with a 1% conversion. This is some of our future work. In the Nature paper, we describe broad photoreceptors, but as you know, there are cones, and we have new data to suggest that we can generate cones. We can generate ganglion cells, which are important for glaucoma. And in mouse, but never in human, we can generate RPE cells. And again, in human, compared to mouse, we can do this even in a week. We thought that it would be harder to reprogram human cells, but it turns out that it's actually easier and it's faster and it's more efficient. So we're really excited by this, and we really hope to take this from bedside to the bedside and have a cell therapy for patients with blindness. And so I'd like to say obrigado, or thank you.
Thank you so much, Sai and Kwon. We were actually amazed by all you have shown us. And we have collected quite a few questions for each of you. We are a bit over time, and I'll try to select some of the questions that the students presented to us. I myself would like just to make a general comment, and this comment is in regard of the biology of the two systems that we gathered today in this session, the eye and the cardiovascular system, and the two profile of researchers which we also had here today. So on the one side, we have Dr. Kwon that has an integrative multivariate approach to the repair of the cardiovascular system, in which he uses different cell types from the pluripotent stem cell, the alleged cardiac progenitor cell, mesenchymal stem cells and their paracrine effect, and combines all of that also with the native or the lyophilized extracellular matrix molecules. And on the other hand, we have Dr. Sai, who is actually focusing all his research and all his bullets on trying to have a cell-free or at least stem cell-free approach to restore vision. I enjoyed both of you very much, and I have myself many questions, but I guess I'm not allowed due to the very many questions you have from them. I'm not very proficient with my fingers, and I have this iPad with me for the questions, but I keep calling one of the organizers to actually help me with, because my sensitivity of the okay, but I know some of the questions. We start by Dr. Kwon, and actually some of the questions that I could gather going through what they showed me are: one, and always present, how far are we from using these in the clinics? The other would be, are your dual combined approach safe? can you actually foresee or envision its use in the combination you are presently using? And another question was, do you believe that this kind of dual approach could be translated into into addressing other diseases such as Crohn's disease? So these were basically the main questions for Dr. Kiwon, who we would like very much to listen, if you may.
Yes, actually, yeah, it's kind of a continuing question after completing this study. This can be applied to clinical situation by using this dual approach. So actually, I would like to try this the obtained result for clinical application, because still stem cell based therapy, as I believe, it's one of the promising direction to regenerate or repair heart failure, but we haven't reached a certain level to use them for clinical situation yet. So we should develop certain reliable and feasible and safe way to use stem cell therapy to repair heart. And our proof of concept study is because we thought that the single cell injection, for example, cardiac myocyte alone is not sufficient to make certain retention and engraftment and survival and make a meaningful contraction to improve heart function. So we thought some additional tool to make let them survive better and engraft better and retention better. So one of the first approach we tried to use is a human mesenchymal stem patch, because we all knew that mesenchymal stem cells secrete a very beneficial paracrine factor to help cardiac myocyte or any cell injected into the heart to make them happy and survive better. So I wouldn't say our dual approach is the perfect system to be able to apply to the clinic, but if we continue to this type of research as a similar direction with our study, then eventually we can make certain direction to make like better cardiac regeneration by using any type of cell. So we are making ongoing effort to use a different cell type or different system or different biomaterial as a tissue engineer approach. So hopefully we can have some good results soon.
Yeah, I believe so. Actually, we have a very integrative approach to the cardiovascular field when it comes to experimental cardiovascular research. And I'm also amazed because in your system, you combine a rat model with pluripotent stem cell derived cardiomyocytes, human, with mesenchymal stem or stromal derived cells from the bone marrow, and with the lyophilized pig extracellular matrix. So you have 3 species together in your in vivo system, and you still have engraftment, okay, and you still get quite a good result when it comes to the modulation of the maturation of the iPS-derived cardiomyocytes, etc., etc. So if you translate directly into the clinic as it is right now, you would obviously, with the ECM, there is no problem, should be no cross-reactivity, devoid of immunogenicity. But would you envision the same kind of therapy going into humans? Would you envision this patch with also the intramyocardial injection of the iPS-derived cardiomyocytes? Would that be a feasible approach into the clinic?
I mean, because we are working hard, so eventually, we can have some good promising result shortly. That's my wish. So that's our ultimate goal. So to do that, we have to make a continuous discussion with the physician, with the several scientists in the different field, like engineers. So I wouldn't be predict that our direction is the final destination, because to keep revising and fix to make a final direction, we just make a continuous effort.
Thank you so much. Just before I let you for this part, what's your what do you foresee that will be a time, if you can, all together in these multidisciplinary teams with bioengineers, biologists, physicians, to have a better approach for heart failure?
Yes, I'm in just like sort of junior level, but we have some other prominent researcher in all over the world, like United States. There are study published in Nature or Nature Biotech using monkey model by using human pluripotent stem cell derived cardiac myocyte. And then in Japan, there is the clinical trial using the cell sheet model using the cardiac myocyte. It's starting this year. And there's some study actually happening in China, trying on human. It's not really clearly reported yet, but we have, I guess, some news that they are ongoing the progress. So by combining all these multiple approach in all over the world, we can have some promising and good result. I hope, yes.
Thank you so much. I agree with you. We are moving forward and at a good pace, finally. Thank you so much. I would now turn to Dr. Sai Chavala, and we have also many questions, although tight on time. And I'll start by, so this I did not memorize as the previous one. So I'll just look at the iPad and try to go through the list. So one of the questions is, have you ever encountered malignant transformation in the cells obtained with your reprogramming? If so, how often?
So the short answer is no. And I think that's one of the huge advantages to transdifferentiation and this kind of cell engineering approach, is that we believe that the rate of malignancy would be extraordinarily small because they're both somatic cells. Now, we do believe that there may be an immature or progenitor type of stage, which is why we can get a diverse population of retinal cells. But we've never seen OCT4 or any type of pluripotency markers other than SOX2, which is really a neuronal marker. We've never seen any pluripotency. So great question.
Yeah, it was a great question, and I would love to be able to see the other questions, which I can't manage to. So the following I would start. The following starts by thanking you. Thank you for your brilliant presentation, Dr. Sai Chavala. And then it should be, do you think there will be and I can't really see, maybe Antonio could help us with, but I have some questions on my own. So I'm very excited with this chemical reprogramming. And a few questions I have on that, because maybe I missed during the presentation, regard the half-time of the cells you are reprogramming in vitro by these chemical signals. So if you start with fibroblasts, at the experimental level, you started with MEVs, they are embryonic cells. If you want to be on the safe side, we are going to use more likely adult fibroblasts, autologous adult fibroblasts, right? So do you have any concern on the previous division history of these fibroblasts for the half-life they will endure after transplanted into the retina? Was it clear, my question to you?
I think so. Were you just asking about the longevity, how long they will survive?
Yeah.
In the retina?
Mm-hmm.
Is that yeah, so neurons are almost impossible to culture. So one thing that I did not mention is that once the chemical conversion process occurs, the cells will die at 2 weeks or 3 weeks. You'll have nothing. And so they will undergo complete death, and only the non-reprogrammed fibroblasts will survive. So that was a big question for us, and that was a big concern. I mean, it was nice that it mimics true photoreceptors, because no one is able to culture photoreceptors or other neurons in culture for an extensive period of time. But that did give us a lot of concern that they would also die in the subretinal space. And from the data that we showed at 3 months when we did histology, certainly a majority of the cells have died. And I would agree with Dr. Ban's presentation where he showed more than 50% would die in a week. We probably had 80 to 90% cell death. But there's a couple of things. In mice, our cell conversion efficiency was low. And so if you look at our peers, they were injecting 300,000 cells into the mouse eye, and we injected 80,000. So, I think there's an easy solution that would be to increase the number of cells. And as I mentioned, in the human system, we can get a much higher efficiency. So that would be more easily achievable. But we do definitely have cells that survive and that maintain at least the cardinal gene, NRL, which is the rod photoreceptor gene, even at 3 months. So.
Yeah, it's brilliant. It's
Thanks.
I'm amazed, although I'm very familiar with the cardiovascular part. I'm less familiar with the eye part, which actually it's a shame, because it's about the second best established stem cell derived therapy in the world after the bone marrow nowadays. But I was amazed with this programming by the single factors, the combination of them. So I think this is outstanding and very promising. Please do.
Thank you very much. I actually have a question, if that's okay, for Dr. Ban.
I believe that's okay.
So are you allowed to ask questions each other, right?
Yeah, I don't know if we're allowed.
No problem, yes.
I would love to marry what we are doing in the eye with the heart, and it goes to what you were describing. What we have noticed, even at day 2 or day 3, is that obviously we're changing epigenetics, of course, correct? I mean, we are changing the epigenetic profile of the fibroblast and really questioning the cell's identity. We saw some beating kind of cardiomyocytes that then dissipate and then they're gone at a very early stage. So is anyone trying this for cardiac chemical approach?
Yeah, there are some study published in Science. It's several years ago, though, using chemical induced directory programming from Chinese group, if I remember correctly. But I never seen any group successful in human cell yet.
Yeah, that would be really cool.
Yeah. So the cocktail of a chemical, small molecule, convert human dermal fibroblast into the cardiac myocyte through, So I think I saw some report, and some people said actually it's not repeated, but I don't know. I haven't tried. Yes.
Yeah.
But direct conversion approach is also very popular in cardiac biology, using several the transcription factors. So from the defoxyribonucleic and others.
Yeah.
Yeah, directly with transcription factors, we have done that a long ago in our field. Deepak has shown that.
Yeah.
But I also recall what Kiwon just said on these. I do not know whether it was a Chinese group or not, but I do recall this. And I do not remember any other work so proeminent in the area about that. So I finally got the other question from the audience to Dr. Sai, and that would be, other than acknowledging how brilliant you were on your presentation, the question was, do you think there will be a way to enhance the survival of the CIPCs in the future, and how?
I believe the simple one is just numbers. So if we understand that there's going to, at baseline, just be a certain amount of cell death, a lot of that is a combination of the procedure itself. So the needle size that you choose is very important because you have to have a small needle because you're making a hole in the retina. And if you make a large hole, then you'll have what's called a retinal detachment. But if it's too small, then you can have cell shearing, and that's where a lot of the cells will die just immediately. And then detaching the retina to create this, what we call bleb, where the cells can go and make these connections is a very traumatic process. So understanding that, we can think about delivering 3 million to 5 million cells, understanding that the vast majority will die, and that might be sufficient to even have a better behavioral response.
Okay. So we are much over time, although I'll just leave a last question for you in case I know it's a difficult one, but I would really appreciate if you could be as short as possible. So we clearly have a huge technical challenge for what you just said, okay? Coming to the size of the needle, the lesion that we are inflicting, etc., etc. So at the biological level, what do you believe is at your specific, at this specific topic, the highest challenge you have?
The greatest challenge is exactly the question that was asked: is cell survival. Because I believe if the cells can survive, then it's not only survival, then it's integration, correct?
Yeah.
So both of those are the key challenges in this field. And this is maybe a higher level question, but experts in the field debate how these cells really lead to vision improvement. And so it may not just be cell-cell synaptic connections. There's cell fusion. And then I think Dr. Ban had described paracrine effects as well. As a physician, to me, it does not matter. All I care about is improving vision. But as a scientist, it is important to understand those differences and to try to exploit them one way or another. We can, one of the things that I have thought about, I think we were alluding to this, kind of bringing it full circle, is in the fibroblast stage, getting the fibroblasts to have a combination approach where we ectopically express or overexpress a transcription factor and then add the small molecules may get us to better cell survival and better cell function.
Yeah, eventually, I would say so. It would kind of help to lock the cells into that state. And
exactly.
Yeah, at least that you would achieve. Thank you so much. We actually had a lot to discuss. I'm afraid we cannot proceed. And I don't know whether the organization has any specific word for the two of you. They do. Please, would you join me? Thank you so much. And from my side, it was a great privilege to be here. And to the audience at home, it's always good to know that you do not give up. And there is a ES meeting every year. Thank you so much to all of you who made it possible.
So,
Pleasure.
we would like to thank you so much for your presentations. We'll now like to unfortunately end our molecular biology session, which was very interesting. We'd like to thank our speakers, Professor Kiwon Ban and Dr. Sai Chavala, for providing us with such high quality scientific lectures, as well as Professor Perpetua Pintuo for her outstanding moderation of this session. So, next up at 3:30 p.m., please do not forget to attend our clinical competition focused on urology. So, we will see you there.
Thank you.
Thank you. Thank you very much.
Bye.
Bye-bye.